Dear students, Assalamu alaikum. Today we are going to talk about our course Molecular Biology, course code BCHM 221. This is lecture number 4 for our online lectures. The topic of today is DNA damage. The outline for today's lecture is we will know about what is DNA damage. We will talk about difference between DNA damage and mutation. Then we will talk about different types of damages that can be caused to DNA. So let's begin with DNA damage. DNA damage is an alteration in the chemical structure of DNA, such as a break in a strand of DNA, a base missing from the backbone of DNA. So in situation where there is some modification in the chemical structure of DNA, or there is some break in a strand, or if a base is missing from DNA, it is known as DNA damage. As you know that DNA work is a software of life. So any change in the sequence of DNA can cause severe ramifications and can result in serious problems. Most of the damages that occur to DNA, they negatively influence the system. So DNA damage is of very serious nature. Therefore, we need to understand the different types of damage that DNA can have, and these damages should be avoided. But in some circumstances, these damages are beyond our control. So we will know about what are the different types of damage that DNA can have. Now, briefly about the difference between DNA damage and mutation. DNA damage is physical abnormalities in the DNA, while in DNA mutation is a change in the base sequence of the DNA. DNA damage can be recognized by enzymes, whereas a mutation, a change that has already happened, it cannot be recognized by enzymes once the base change is present in both DNA strands. In this situation, if the change is present in both the strands, this change will be carried on along the different replication stages, because in both the strands, it means the parent strand, when the two strands will be separated, the parent strand will carry this change with itself. So the system will not be able to recognize it, and this change will be carried to the next generations. So this in mutation, it can't be recognized once a mutation has established itself. DNA damage can be correctly repaired if redundant information, such as undamaged sequence in the complementary DNA strand or in a homologous chromosome is available for copying. So there is a possibility of this damage can be repaired if the complementary DNA strand or in a homologous chromosome is available for copying, while a mutation once happened, the mutation cannot be repaired, because as I told you before in this part, it becomes part of both DNA strands. So now the system will not be able to repair it. Now there are different types of damages to DNA, and that can happen. One of those types is single base alteration. There are various examples of single base alteration. One of those examples is depurination. Another example is deamination of cytosine to uracil. Another example of single base alteration is deamination of adenine to hypoxanthine. Another example is alkylation of base. In another cases, insertion or deletion of nucleotide can happen in single base alteration. And in another case, base analog incorporation can possibly happen. So these are the different types of single base alteration. All these different types are example of single base alteration. We will study in detail about some of these examples, and it will give you an idea of how single base alteration can possibly happen. Then another broad type of damage to DNA is two base alterations. It can be caused by UV light induced thiamine, or this is called pyrimidine dimer, or it can be by bifunctional alkylating agent or cross linkage. Another type of DNA damage is chain breaks. Chain breaks can be caused by the examples of these are ionizing radiation can cause chain breaks, radioactive disintegration of backbone element it can cause chain breaks, and oxidative free radical formation can be involved in chain breaks. So these are the different examples of how DNA damage can occur. Another broad category is cross linkage. The example of this is between bases in same or opposite strands, cross linkage can be formed. And another example is between DNA and protein molecule, for example histone, this cross linkage can be formed. So this table basically summarizes the different types of DNA damage that can be caused by different agents, and these are the different examples. We are going to talk in detail about some of these examples. So let's talk about first single base alteration. The example of this is depurination. It is caused by most of the time it's caused spontaneous. It can be chemical induced like estrogen. It can be UV radiation. It means UV radiation can cause this situation. Ionizing radiation is another example which can cause single base alteration. Monofunctional alkylating agents can be responsible for this, and free radical is also an example. So another one, deamination, hydrolytic removal of an amine group from a molecule. Its causes can be spontaneous, or it can be chemically induced like nitric oxide induced deamination. This is example of how spontaneous deamination can possibly take place. This is, you see, cytosine, and with this deamination it can be converted to uracil. Adenine through the process of deamination can be converted to hypoxanthine. Guanine in the process of deamination can be converted to xanthine. 5-methyl cytosine can be converted to thiamine. So these are the different examples of how deamination can take place. In one can see that cytosine converted to uracil, then converted to hypoxanthine, guanine converted to xanthine, 5-methyl cytosine converted to thiamine. Alkylation, alkyl group seen H2N plus 1, methyl ethyl propyl, transfer of an alkyl group from one molecule to another. It can also cause DNA damage. There is another example which is called base analog incorporation. Base analogs become incorporated into daughter strand during DNA duplication. For example, 5-bromo uracil is a thiamine analog. It can be incorporated into DNA instead of thiamine. You can see here 5 bromouracil, its keto form, and the normal pairing with adenine. This tautomeric shift occurs at a relatively high rate. 5 bromouracil enol form, mispairing with guanine. This is guanine, and this base analog incorporation can take place, resulting in damage to DNA. Mismatch base can also possibly function in DNA damage. Cell makes mistake for recognizing the bases due to poor proof reading function. Sometimes causes change in base. For example, G could look more like an A. So this situation, normally you know that A binds with T and G binds with C, but when G could look like more like an A, this can cause problem, and this can cause mismatch base and can cause damage to DNA. Now in some cases we can have double base alteration, like pyrimidine dimer. This is caused by UV radiation. It can maybe direct effect of UV radiation. This is an example of pyrimidine dimer. One can see here thiamine, and this is another thiamine. Through UV radiation, there is a possibility of formation of this thiamine dimer, and this thiamine dimer results in 2 thiamine binding each other. This can cause DNA damage because it is now not normal situation where adenine binds thiamine and guanine binds cytosine. Here one can see formation of this thiamine dimer. So there is a possibility of UV induced DNA lesions. They can be through pyrimidine dimer. They can possibly be through purine dimer, and can occur through the process of depurination. It can occur through single and double strand break. Now another way in which DNA damage can occur is chain break. This chain break can be single stranded break. It is caused mainly by irradiation, or free radical damage can also result in single stranded break. Double strand break can also happen, and causes, there are different causes of this. This can be caused by direct by irradiation, or it can be caused by unrepaired UV induced DNA lesions. It can be caused by the repair of single strand breaks. It can be caused by free radical injury, and radiomimetic compound can cause double strand break. Now free radical damage to DNA. Now let's first talk about free radical. As you know that in our body there are different types of reactions happening all the time, resulting in generation of free radicals. These free radicals, they are highly reactive, and their highly reactive nature can cause damage to DNA, and these free radicals are normally scavenged by antioxidant system present in our body, but sometimes they in cases of oxidative stress or Another situation, when the free radicals are not properly scavenged, they can cause damage to DNA. So this is why the free radical can cause damage. Oxidative damage mainly occurs by formation of free radical or radiomimetic compounds. Free radical damage to the DNA is 70% damage by OH. Radiolysis of H2O produces peroxide. Formation of OH requires metal near to the DNA. Fe+2 mainly forms OH. Formation by Fenton reaction. Here you can see the hydrogen peroxide and the OH radical formation. These OH radical having this potential of or taking anything can cause damage to DNA. So these free radicals, they are very highly reactive and they can cause damage to DNA. There are different types of DNA and damage that can be produced by free radicals. Some of these are depurination. That means that free radicals can perform the depurination of the bases, and depurination of the bases will alter their structure, and this will result in a DNA damage. And then the free radical also have the capability to break single strand. They can cause single strand break. The free radical damage is not only limited to single strand break. Free radical also have the capability to cause double strand break. So these are the different types of damages that free radicals can cause to the DNA and resulting in DNA damage. The DNA damage that can be caused by all these different methods are not left unchecked. There are proper DNA repair mechanisms in the body, and thanks to those DNA repair mechanisms, that these different types of damages that can occur to DNA, they are, in majority of the cases, they are repaired. Because if they are not repaired, it can result in many complications. So in the next lecture we are going to talk about how these different types of damages that can be caused to DNA are repaired. Because as I told in the introduction of this lecture, that DNA is the software of life. It is very important for transmitting the information that it is stored in it. So if there is any damage to DNA that has not been repaired by the DNA repair mechanism, it will have severe ramifications. So DNA, this is why, and after understanding the DNA damage, different methods of DNA damage, we will talk about DNA repair mechanisms, how DNA is repaired through different processes. So I hope once you have the idea of DNA damage, it will also give you the importance of DNA repair mechanisms. So I hope you understood about the different methods through which DNA damage can occur. And then now from here we move towards DNA repair mechanism. Hopefully you enjoyed the lecture, and if you have any questions, please feel free to let me know. I will be very happy to answer your question. Thank you very much for your attention. And this